Hello everyone, my name is Brian, and I'm from the Duke Center for Combinatorial Gene Regulation under Tim Reddy. And I'll be going over a quick demo of the Ccgr portal. We view the portal as a complementary piece to the puzzle. Building on the foundation of catalog-style databases that store experiment data like IGVF and ENCODE, the portal can take the next step and focus on the results of these experiments. We pull experiments from two major classes of functional characterization assays, high-throughput reporter assays and perturbational screens. This enables users to easily access and explore a variety of analysis linking genes to regulatory elements. Reduce your workload and save time. Researchers typically spend large amounts of time digging through experimental results, parsing data, and formulating it to find the answers they need. Our portal is designed to do some of that work for you, streamlining the process and allowing you to focus on your research. Since the portal is already integrated with CCREs, gene information, and merging capabilities, that helps make data exploration more efficient. Ultimately, we view the portal as a way to share information across consortia. There are a few key features we'll explore in the portal. Number one, find regulatory effects targeting your gene of interest. And this allows us to discover regulatory elements controlling the expression of your gene of choice. Number two, find, compare, and visualize regulatory activities in your regions of interest across chromosomes. And number three, upload data to visualize and compare with surrounding regulatory activity, making your research clearer and more insightful. Let's say you want to discover regulatory effects for your favorite gene. You can search by gene symbol here. Since we already have HLA populated, we'll click Discover. And this will take you to the gene's profile page. HLA up top. If you scroll down, we have the Genome Browser. The Genome Browser shows all significant hits found in this region around HLA from the data uploaded to the portal. To the left, you'll see Screen CCREs, all tested elements, reporter assay hits, CRISPR-A, CRISPR-I, candidate enhancer promoters, repressors, and then we have the HLA gene below. If you have any questions regarding any type of terminology, if you scroll up, we have a glossary button at the top that you can review if you need any assistance. As you scroll down, we have our tables. Right here, the targeting regulatory effects observations. And this is basically regulatory effects observations targeting HLA-A. And to the right, we have download buttons for TSV and BED6. If we scroll up and type in LMO2, for example, another gene, there's another tab, Proximal Regulatory Effects, and these are nearby regulatory effect observations that have not been experimentally linked to LML2 or any other gene. And this data is coming from experiment data like proliferation screens and attack star seeds. As we go back to the home page, let's say you want to compare results between experiments and select and analyze multiple experiments of your choice. We can click the List Experiment button. Now we're at the experiment index page. Filter experiments to the left. Let's grab a couple of experiments. You can either click the plus button or drag and drop if you like. Click Analyze to selected experiments. And this will take us to the multi-experiment coverage information page. You can select whether or not you want the intersected data or the union data. Now we have a chromosome-wide view of the coverage of both of these assays. You can filter the data to the left. And if we take a look at the chromosome map, the top is the tested element effect significance, and the bottom are significance of effect on assay genes. As you see, we have highlighted regions on the chromosome map. The top regions, like here, represent tested elements, the region where the tested elements are located. The bottom regions, the bottom highlighted regions, are where the genes are located. What's unique about this is if you hover over the top regions where the tested elements are, we can see the corresponding linkage between the genes below. If we hover over a bottom gene region, it'll show the same thing, which is the linkage to the tested element in which it's being influenced by. If you have a bed file, you're able to use it and overlay your data on top of this current experiment data. If you're logged in, you can download all of the data or some select regions of your choice. Taking it back to the home page, Some may want to summarize results for a genomic region. You can search by region here, click show, and this will show the hits from all of the data that's within this region, all the significant data that's in this region. We have the summary results. So for an example, we have within this region 2 genes, right here, LMO2, this ensemble gene right here. Top significant regulatory effects observations. So these are the top 5 significant regulatory effects observations for each experiment that covers this region. We click on the all effect tabs. So this is all of the significant regulatory effects observations found in this region. And again, we can download TSV Bed 6. You can also upload your data to the portal to visualize and compare with surrounding regulatory activity with the data that we already have. Right now, you can email us and we can upload your data, but soon we'll have that interface for upload accessible to users. Thank you for stopping by, and we hope to see you soon. Thank you.